Hi, I'm Mary McQuilkin, adjunct faculty for the University of Alaska Fairbanks Center for One Health Research. This video is 3 case studies. The topics I selected each have examples of successful mitigation strategies that were implemented, but they'll each require ongoing surveillance and would benefit from additional work to address the root causes of these threats to human, animal, and environmental health. Each case is followed by a set of 6 questions, 2 focused on humans, 2 focused on animals, and 2 focused on the environment. You'll discuss the questions with your team for the second assignment and submit a summary of your team's responses. More details can be found on Canvas. The first case study is harmful algal blooms, which is also the topic of the interview with Gay Sheffield and Rick Toman. Harmful algal blooms is a topic we'll revisit later in the course, but I wanted to go ahead and introduce it here because it's an important emerging threat in the Arctic at the intersection of human, animal, and environmental health. Saxitoxin is a neurotoxin naturally produced by some marine dinoflagellates, including Alexandrium species. Warming ocean temperatures around Alaska have increased the frequency and geographic range of harmful algal blooms. When saxitoxin accumulates in shellfish, it can cause serious illness or death in animals that consume it, called paralytic shellfish poisoning in humans. Warmer waters result in a rapid buildup of harmful algae populations. As a result, Alaska's seabirds and marine mammals are being exposed to increasing levels of toxins. Symptoms in people include numbness of the lips, mouth, face, and neck, muscle weakness, lightheadedness, ataxia, motor incoordination, drowsiness, incoherence, progressively decreasing ventilatory efficiency, and in high doses, respiratory paralysis and death. People start to experience symptoms from minutes up to about 4 hours after eating contaminated seafood. Paralytic shellfish poisoning has a case fatality rate up to 12%, with death occurring 2 to 12 hours after seafood ingestion. For those who survive, symptoms start to improve after 12 hours and resolve within a few days. Dinoflagellates are a type of phytoplankton that can be a food source for certain zooplankton. For this example, let's say the dinoflagellates have a concentration of 2 micrograms of saxitoxin per 100 grams weight. When zooplankton consume those phytoplankton, the concentration of saxitoxin in their bodies increases to 10 micrograms. As filter-feeding shellfish like mussels, clams, and oysters consume these organisms, the toxin accumulates in their tissues without it causing significant harm to them. As these contaminated shellfish are eaten by other organisms in the food chain, like fish, marine mammals, and ultimately humans, the saxitoxin can build up to higher concentrations at each trophic level. While there isn't a universally established threshold for saxitoxin in marine mammal meat, the FDA has set a regulatory limit of 80 micrograms of saxitoxin equivalents per 100 grams of shellfish meat. Exceeding this limit in commercial shellfish leads to harvesting closures and advisories. This is an additional risk for rural Alaska Native communities where there often are no regulatory agencies to post health advisories. I made this illustrative example not using real data, but I wanted to show the general idea of how the concentration of saxitoxin increases going up the food chain. The paralytic shellfish poisoning monitoring and response effort is an example of multidisciplinary collaboration between tribal, federal, state, and community stakeholders. The program has been successful in reducing the risk to human populations while gathering data to inform measures to protect the ecosystem. What human behaviors may have contributed to this problem? What policies or behavior changes might reduce the risk or alleviate the problem? Describe the changes to animal population health related to harmful algal blooms. What species are you most concerned about protecting from toxin exposure? How has ocean warming affected harmful algal blooms? What additional environmental changes could be interacting with ocean warming to influence bloom dynamics? Avian influenza is a highly contagious viral disease, first described in northern Italy in 1878. It was known as foul plague before the causative agent was discovered to be a virus rather than bacteria in 1955. In recent years, the epidemiology of highly pathogenic avian influenza A virus has changed, with it becoming endemic in domestic birds in a number of countries and causing major outbreaks among domestic but also wild birds worldwide. It's transmitted through the respiratory secretions and feces of infected birds, which can contaminate water or feed, enabling it to spread efficiently through large flocks. It can be spread by the movement of birds themselves or on surfaces such as agricultural equipment. It's classified as either low pathogenicity avian influenza or high pathogenicity avian influenza. Low path avian influenza may cause minor illness, but sometimes causes no symptoms at all. But high path avian influenza causes severe illness with high mortality rates. Highly pathogenic avian influenza virus A, H5N1, was first detected in chickens in Scotland in 1959. Many decades passed, then in 1991, H5N1 was identified as the cause of an outbreak among turkeys in England. In 1996, it was identified in goose farms in southern China. A year later, that same viral lineage caused an outbreak in live poultry markets in Hong Kong, which resulted in 18 human cases with 6 fatalities. Over time, highly pathogenic avian influenza A, H5N1, has continued to evolve and adapt to multiple hosts. In humans, it caused 868 infections across 20 countries from 2003 to 2023, with a case fatality rate of 56%. Since 2005, highly pathogenic avian influenza A H5N1 viruses have undergone extensive genetic diversification, including the formation of hundreds of genotypes following reassortment with other avian influenza A viruses. After clade 2344b was detected in North America in 2020, it spread to Central and South America, and by 2023, it had reached Antarctica. More than 30,000 South American sea lions died as the H5N1 virus spread along the coast of Peru and Chile in 2022, with porpoises, dolphins, and otters also being affected in smaller numbers. The virus made its way down the Chilean coast in 2023, then spread up the east coast of Argentina onto Uruguay and Brazil. In October of 2023, over 17,000 elephant seals died from the virus during the largest mass casualty event ever recorded among elephant seals in central Patagonia, Argentina. The pups in the Punta Delgada colony that year suffered a 95% mortality rate, compared to 1% in prior years. There was a concurrent outbreak in terns locally, with peak mortality 3 weeks later. Observers documented disoriented terns with neurological symptoms, many were struggling to fly or unable to fly, and dead terns were mixed with elephant seal carcasses covering the beach. Other outbreaks in mammals include farmed foxes in Finland, farmed mink in Spain, and pet cats in Europe. Highly pathogenic avian influenza A H5N1 was first detected in dairy cattle in Texas in March 2024, among 2 domestic dairy cows and 2 cats from the Panhandle region of northern Texas, where dead wild birds were found near the farms. That year, infections in U.S. dairy cattle were confirmed in over 80 farms across 9 states, with farm-to-farm spread. Infections in mammals have included a polar bear, harbor seals, gray seals, baby goats, alpacas, and domesticated pets, including cats. In the U.S., the USDA Animal and Plant Health Inspection Service, which monitors for avian influenza A viruses in wild, commercial, and backyard birds, reported virus detections in more than 9,300 wild birds in 50 states and territories, and more than 1,140 commercial and backyard flocks, affecting more than 96.5 million birds in 48 states from 2022 to 2024. The current outbreak of clade 2344b, which began in January 2022, is the largest and longest in U.S. history. Major outbreaks have occurred in nearly every U.S. state among birds, mammals, dairy cattle, and humans. While no cases of human-to-human transmission of highly pathogenic avian influenza A H5N1 virus have been identified, there is a risk of additional spillover to humans going forward, both because of the potential for influenza viruses to rapidly evolve and the high global prevalence of this virus in domestic and wild animal populations. To mitigate risks and guide preparedness, ongoing surveillance in birds, animals, and people remains critical. Beginning around 2003, a deadly strain of H5N1 avian influenza started spreading widely in Asia, but mostly impacted domestic poultry. Vietnam was one of the most severely impacted countries. In 2003 and years following, 44 million chickens died or were culled to prevent the spread of virus in Vietnam. There were 119 confirmed cases in humans, with a case fatality rate of 49.6%, which was 59 human fatalities. To control the outbreak, leaders from the health, agriculture, and environmental sectors formed a committee. They worked together to guide the response and developed a system for early warnings and future outbreaks and for outbreak prevention. After an initial regional grant-funded effort ended, in 2005, to continue and expand avian influenza prevention and control efforts, the World Organization for Animal Health and Food and Agriculture Organization of the United Nations formed OFLU. Their network continues to gather and share international surveillance data on highly pathogenic avian influenza and is applying a One Health approach through collaboration with international partners on mitigation and control efforts. Ongoing work remains critical to address highly pathogenic avian influenza, so this isn't a story with an ending, but it does provide an example of One Health in action to mitigate a global threat to human, animal, and environmental health. Explain how farming practices and trade patterns may have facilitated the spread of H5N1 in 2003 and similar outbreaks since then. What public health, veterinary, or agricultural measures could people have taken in advance of outbreaks to reduce the risk of occurring? Why is highly pathogenic avian influenza an ongoing risk to animal populations today? Which animal populations are most vulnerable to influenza outbreaks and why? How might climate change or seasonal patterns influence H5N1 outbreaks? What other environmental or economic factors can contribute to the transmission of H5N1 and similar viruses? In 2006, a spelunker was exploring Howe Cave in New York, and he encountered bats with a strange white fuzz on their faces. A year later, biologists conducting a routine bat count in Albany, New York, found the same scene. Bats had a white fuzz on their noses, with some spots on their ears and wings. Where there used to be thousands of bats hibernating on the ceiling, they instead found the cave floor littered with bat carcasses. The fungus was identified as Pseudogymnoascus destructans, or PD, and it quickly spread to other caves in the region, then other states, and reached the West Coast in 2016. Bats in 40 U.S. states and 9 Canadian provinces have been confirmed to have white nose syndrome. About 6.7 million bats have died of white nose syndrome since 2006. This map is color-coded to show the spread of PD from 2007 in white up to 2025 in red. Over 90% of bats in some areas died when the fungus arrived, and several species are now at risk of extinction. The fungus Pseudogymnasticus destructans can thrive in cold conditions like the caves where bats hibernate over winter. It infects their noses and skin, and it can spread from bat to bat through skin-to-skin contact as they cuddle up against each other while hibernating. As it infects their skin, it causes them to wake up from hibernation and exhaust their energy stores. So many leave their cave before spring, desperate for food, but long before the insects are back. Piles of dead infected bats have been found in the snow. After the outbreak was identified in the US, biologists tested for it in other parts of the world, and they found that bats in Europe and Asia have Pseudogymnasticus destructans, but it isn't causing harm in those populations, likely because they've adapted to it because it's been there for a long, long time. It's likely that the fungus was brought to the US by a traveler by mistake. People can spread fungal spores on their shoes, clothing, or outdoor gear by accident. I was going to summarize the research on this, but then I found a great lecture by wildlife biologist Dr. Tina Cheng of Bat Conservation International presenting her own research. So I'm going to include a couple clips from her presentation.
This disease has had catastrophic impacts on a few bat species. One of them is a little brown bat, which was once the most common bat in North America. And as you can see in this animation, the gray dots represent the spread of the disease, and those red circles that were big, this is before white nose hit. And then you can start to see in 2006, the size of those red circles, which represents the size of those bat populations, are starting to shrink down, representing the mortality caused by white nose syndrome. We examined these population counts in 5 species of hibernating bats in this recent publication that we worked on in collaboration with the North American Bat Monitoring Program and US Fish and Wildlife Service. And here I'm just going to show you the top 3 species that have been the most severely impacted. Here on the left side, you can see these maps. The gray zones represent the distribution of the species. The red space represents where WNS has been confirmed. And those yellow dots are all the little hibernacula, all the little sites that have been monitored over time. This is a monumental effort by lots of state biologists all over North America that came together to bring their data together so we could understand how white nose syndrome was affecting bat populations from these species. What we're representing in these top panels are the actual counts. This is in log scale, so it's a little bit confusing. But here, the 3, 1e to 3 represents thousands of bats. Okay, if it were 1e to the 4, that's tens of thousands of bats. So some of these colonies were really huge. They literally could be tens of thousands of bats at a single site. And so this little dot here represents like the average number of bats that you would find in a site. And you can see that pre-arrival, so before white nose syndrome came, this is what the bat populations were estimated at. And then invasion were those first 2 years since PD came. And you can see that already these bat populations are starting to drop, they're starting to decline. And so in this bottom graph, what we're showing here is that change between each disease stage and that pre-arrival point. So in that first year of invasion, you can see that this bat, Myotis septentrionalis, also called the northern long-eared bat, has already declined by greater than 75% just in those first 2 years. As the disease progressed from the first 2 years, the invasion stage, to what we call the epidemic stage, that's years 2 through 4 following pathogen invasion, and then established as years 5 and on. So, what is the long term impact of this disease? And if we just focus on that established stage, you can see that it is really severe for these 3 bat species, close to 100% for northern long-eared bats. So, in many cases, WNS was hitting these sites and causing those bats to be completely extirpated or completely wiped out from that site. Little brown bats and tricolor bats also very severe declines, greater than 90% following the invasion of WNS. And so, for this reason, these species have in the US gone through a review for protection from the Endangered Species Act. The most recent ruling ruled the northern long-eared bat as an endangered species, which means that based on the data and information we have, we believe that this bat may be driven to extinction solely because of the invasion of this novel pathogen and the impacts of this emerging infectious disease. Why is white nose syndrome so deadly? It is an introduced pathogen affecting naive hosts. And this has been shown in other systems to be a really dangerous trait. You have a pathogen that maybe has evolved, like for in this example, in Eurasia over a long period of time and has evolved to be really hyper virulent, but in the hosts for which this pathogen is endemic, they have evolved with it. So they have some sort of defense mechanism. But when that pathogen then gets displaced and introduced to hosts that have never seen it before, it can be really hyper virulent, can be really deadly to those species. Another thing that makes it so deadly is the timing. This is a time in which bats are hibernating. And hibernation means that they're shutting down a lot of their bodily functions, including their immune response. So they're really vulnerable during this time. Bats are able to make it through the winter because they go through what we call torpor. They actually bring their body temperature all the way down to close to the ambient temperature of their surroundings. And they slow down their metabolic rate. So they're really not expending much energy at all. They actually use 80 to 90% of their actual energy budget arousing from torpor. There are some scientists that cleverly put temperature loggers on the bats themselves so it can measure their body temperature throughout winter. And so, what we have here on the x axis is November through March. So, this is their hibernation time. And on the y axis is their body temperature measured by that tiny little temperature logger that they put on the bats. And you can see they are staying like 10 degrees or below. So, this is probably what the cave temperature is measuring at, or very close to it. But what are these big red spikes? These are their arousals. This is when the bat is actually heating up their body from like 10 degrees Celsius to like almost 23 degrees Celsius. And that takes a ton of energy. So you can see that this bat, this is a healthy normal bat, is arousing every periodically, every so often. And it's not staying up for very long, just like a couple hours. But this is when they really save their energy through that torpor period. And that's what gets them through winter. What happens with WNS bats is that they are perturbed by this fungal infection on their skin and they arouse much more frequently, twice as frequently as a healthy bat. And you can see here just how dramatic that difference is. And you can imagine every time it's waking up, it's burning through critical fat reserves. This is thought to be one of the main mechanisms towards death is that they burn through the fat reserves and starve to death before they're able to make it out of winter. Another reason why this pathogen is so deadly is that it is a multi-host pathogen, which means that there's lots of different species that it affects. There's actually 12 different bat species that have been confirmed with whiteness syndrome, another 6 species where we detected the pathogen, but they don't develop any of the symptoms of the disease. And so when you have different species with different types of susceptibility, it becomes very dangerous for those hosts that are very susceptible to it. You can have sort of a typhoid Mary situation where there's a bat that has a pathogen and is totally fine and it's just spreading it around and keeping it within the system and not allowing those other more susceptible hosts to escape it. The other way that they contact it is from the environment. They drop fungal spores on the cave or subterranean habitat where they're hibernating. And because it's a fungus, it can live off the host. It can be incredibly persistent. Here's an example of that. A colleague of mine I did my PhD with, Joe Hoyt, he was there in the very beginning when PD first hit in New York. And he was working in a lab where they cultured the fungus from bats. And this is the original culture up here on the top. And what happened though is that they cultured this fungus, they put it in the fridge, but then that lab shut down. They turned the fridge off, they unplugged it. He came back 5 years later, opened up the fridge, found this dried, caked out petri dish with the fungus, and decided to put it onto fresh culture to see if it was still viable. And he found that it was. This is an incredibly persistent fungus. There have been other studies that have shown that the fungus can persist even after the fungus has totally killed all the bats off in the cave. They've come back a few years after, swabbed the cave walls, put it on a petri dish, fungus comes back to life. This also makes this pathogen very deadly. You don't even have to come up against another susceptible host. You can just rub up against the surface where PD is just sitting there waiting for you and be infected by it and succumb to the disease. This is a clonally reproducing pathogen, very different from a lot of viruses that actually go through tons of mutations. It does have the ability to reproduce sexually, and we know that that exists in the European strains. But this strain that is spreading through North America is spreading through clonal reproduction. And this turns out to be one of the mechanisms that some pathogens potentially use to keep their virulence, to maintain that hypervirulent stage. Another interesting side note about PD is that it glows in the dark. This is actually the fungus that is fluorescing orange. It produces this phosphorescent protein when it is actively infecting skin cells. Don't know why, just a property of the fungus. But it provides a useful tool for surveying and seeing whether or not bats are being affected by PD, whether it's actually digging into their skin. We oftentimes do see the visible fungal growth, but here's an example. This is a little tricolor bat that we found, and you don't see any visible fungal growth on it, right? No white spots. But when you shine the UV light, you can see these orange spots. This turned out to be another characteristic that we didn't always see white fuzzy fungal growth, did not mean that the bat was not infected by the pathogen. And in fact, a lot of times you don't see the white fuzzy fungal growth, but you will, under UV light, see that the bat is highly infected with the pathogen. This fungus is not the only thing that glows in the dark. There are other microbes that also do this. And one of them is this bacteria called Pseudomonas fluorescens. It actually, its name was given to it because it has this property that it glows blue under UV light. Here's another bat. You can see that its wing is just completely destroyed by PD. There's orange spots all over it. It's also glowing blue. And here's a petri dish. Here's what the bacteria looks like to the naked eye. And then here's when you turn a UV light on, you can see it glowing blue. If you look closely, you see these little orange dots, but around it, there's no blue. And that made me think of what we often see in the petri dish when we put bacteria that has antifungal properties on a plate with the fungus. I'll make the disclaimer, I don't actually know if this is Pseudomonas fluorescens. We never tested for that. But just to give an example, that there are microbial interactions that could potentially naturally be happening on bats in the wild. The question is, could we then use this to our advantage? Could we figure out what are the microbes that are outcompeting PD and develop it as a probiotic to go back and give bats a big dose of this so that they could better fight off the fungus on their skin? This was work that we did, led by Joe Hoyt, my colleague at UC Santa Cruz during my PhD. We started with this lab experiment where we actually went into the field, swabbed bats, cultured out Pseudomonas fluorescens from bats, and then did what we call these challenge experiments in the lab. On the left here is a plate that is just PD only. So this is what the fungus looks like if it's growing uninhibited. And this is a plate where we spread PD on it, but then we did 3 drops of the bacteria. And what you see, this yellow stuff is the bacteria growing. And then the white zone around is what we call the zone of inhibition. So that's where it's sending out these chemicals, these antifungal chemicals. And the fungus does not grow around the bacteria because it is able to fight off the fungus. So this is very encouraging evidence that potentially we could develop Pseudomonas fluorescens as a probiotic tool to fight off PD. We tried really hard to see if we could develop this tool, but we ran into a lot of challenges and a lot of issues. Following that first initial lab experiment, we then did a captive bat experiment, brought bats into lab, sprayed them with the probiotic, challenged them with the fungus to see if it would help bats survive. And where we found this really encouraging zone of inhibition in the Petri plate, we only were able to detect a 30% increase in survival for bats that were treated with the probiotic. It is so difficult to do both captive bat treatments and wild bat treatments because of the nature of hibernating bats. They are very sensitive during this period of time. So any disruption to them has an impact, but also because especially for wild bats, if you treat one, you have to be able to catch it at the end to know if there was efficacy in treatment. And being able to do that was just incredibly challenging. I'm going to talk about a little bit of these other treatments that have gone through the pipeline. Another one's called Rhodococcus rhodocris, which is a bacteria that was the brainchild of Chris Cornelison, who was working in a lab trying to figure out how to prevent bananas from getting moldy. It turns out there's a bacteria, and it's this little orange guy here in this Petri plate called Rhodococcus rhodocris, RR, I'm going to call for short, that produces volatile organic compounds. So these are now aerosolized chemical compounds that have antifungal properties, and you don't even have to spray the bananas. They just put it in the same Tupperware as the bananas, and it helped the bananas to prevent ripening and fungal growth. So Chris thought, well, could we do this with PD? So he did a lab experiment. Here's the 2 Petri dishes. On the left here is PD only. You can see white fuzzy, as we expect, uninhibited. Here's PD on the left side, and here's Rhodococcus on the right side. They're not even touching. They're just in the same dish together, and there's no white fuzzy fungal growth on there. Just the presence of Rhodococcus next to it was able to inhibit growth of PD. Pretty cool. There was also another technology. Dan Lenner and his colleagues found that you could expose the fungus to just ultraviolet C light and it would kill it. And the special property of the fungus thought to be indicative of its co-evolution with bats in subterranean environments is sensitivity to UV light. What you see here is their challenge experiment in the lab. And across the top, here's the control. So this is no UVC treatment. And then as you move from left to right, increasing amounts of UV light shown onto the Petri plate, then different fungal strains, but don't worry about that. Really, what you can see here is that as you increase the amount of UV light treatment, you get less and less fungal growth. And towards, the last two treatments, like none. You can prevent PD from growing, with the exception of this little guy here. Let's call him an outlier. They put Petri plates around a room to see if you could pull in this portable UV light, put it into a room, and turn it on for a couple hours. And if it would reduce fungal growth, and they found that it had 97% fungal reduction using this whole room sanitation method. And of course, there's a vaccine. This is a vaccine that is being developed specific to PD. And something unique about it is that there really are not a lot of fungal vaccines that have been developed. It's a breakthrough for PD treatment, but also just for medicine in general, trying to figure out how do you create a fungal vaccine. It's very interesting. The way that they do it is they call it viral vectors. So they actually try to figure out what are the things that a host will respond to, these antigens that their immune system can identify as PD being a pathogen and something that their immune system can then be like, on the attack, we recognize you're an invader, and now we're going to mount a mass immune response to kick you out. And so they're able to get these special antigens into viruses and then use that as a way to mount an immune response in bats. The other cool thing about it is we get our injections of our vaccines, and they did try that. They did try a injected vaccine. They also were able to just spray it onto their noses, and the bats were able to get the vaccine by ingesting it orally, which makes it much more logistically feasible. What I'm showing you here is a figure from their paper where the blue is the control. So they got no vaccine. On the x-axis is how many days that those bats survived and the percent survival on the y-axis. So you can see for the bats that did not get any vaccine, they went through hibernation for some period of time, but at some point, the survival dropped down to, I don't know what that is, about 30%. So those bats were tanking. Now you see the red and the green. These bats all got vaccines. The red got an oral vaccine, the green got injection. And you can see that they survived much better. They still, at some point started to have some impacts, but there's a significant improvement in survival because those bats were vaccinated. I gave you an example of just a few things that have been under development. There have been a lot of different tools and strategies that have been trialed in the lab, that have been trialed on bats in the lab, that have been trialed on field experiments. But sadly, we have not been able to get to a point where we have demonstrated enough efficacy to actually deploy anything at scale. These are population counts of little brown bats in the northeastern US. And again, just to help you understand this log scale, 3 is like thousands of bats, 4 is tens of thousands of bats. And you can see over time, there were some really large colonies. And then in 2006, 2007, white nose syndrome hit, populations start to tank. But there are some populations that tank and then flatten out. This was the first indication that there may be some populations that are figuring out how to persist even with the disease. We have seen populations now that are persisting with lower fungal loads. So something is fighting off the amount of disease as on individuals. It seems like bats are also roosting in colder microclimates where PD may not grow as well, but also where they're able to regulate how often they arouse from torpor. Bats that roost in colder microclimates arouse less frequently. So they're regulating their hibernation energetics in such a way that they can make it through winter. And then we found that there are bats that have increased the amount of fat that they come into winter so that they're able to tolerate all these extra energetic impacts. And this is what we call the fat bat hypothesis. This was actually first observed by a state biologist, Greg Turner, in Pennsylvania. he was catching bats and noticing that they were much heavier than they used to be. One of my dissertation chapters actually followed up on this hypothesis, went out to several sites where there were persisting little brown bat populations and measured their body fat. And I was able to compare it to body fat of bats at those sites before WNS had caused mass mortality. And what we found is that at 4 of the 6 sites, bats had significantly more body fat. And so what you see here in this figure is the percent body fat. The blue curve here is the distribution of body fat in bats before white nose syndrome impact. And you can see the green curve here shifts it over to the right. So now where bats used to be like 15% body fat, now they're about 30% body fat. So they're coming into winter with greater fat reserves. And we estimate that that could reduce their white nose syndrome mortality by up to 64%. Something that we're currently working on is the idea of whether or not we could help bats to increase their fat reserves in the fall before they go into winter, and also when they're coming out of winter hibernation and about to go into the breeding season. As a pilot, we set up these UV lights. And as you probably know, whenever you go out and you see a light, there are bugs that will just congregate at them. So we created these bug buffets and we placed them in our hibernacula to see if bats would use them. And the short answer is yes. If you create these bug buffets, bats do come and they increase their foraging activity around these lights, and that may actually help them to increase their fat reserves. Now, in the long run, we're really hoping to do habitat restoration rather than putting up UV lights to help bats be more resilient, not just to white nose syndrome, but all the other external pressures that they are handling on top of that. It's going to be a long road to recovery, but we haven't given up and we are still looking for ways to facilitate recovery and resilience in populations affected by white nose syndrome.
If you were watching Tina Cheng's presentation while thinking about how much you hate bats, bats can eat 40% or more of their body weight in insects per night. A single bat eats 1,000 insects in a night, and some larger species can eat up to 8,000 insects. This includes not only backyard pests, but also many insects that damage crops, so bats are important for farmers and for our agricultural system overall. A 2010 study valued bats' pest control services in the United States at up to $53 billion per year. Research by Frank, published in 2024, found that on average, farmers increased their insecticide use by 31.1% in areas where bat populations had been killed by white nose syndrome. He then looked at infant mortality rates and found that counties with white nose syndrome in their bat populations had an infant mortality rate 7.9% higher on average compared with counties whose bat populations did not yet have white nose syndrome. This higher infant mortality rate meant 1,334 excess infant deaths. This research was uniquely able to establish evidence for a causal relationship between increased pesticide application and higher infant mortality. Causal relationships, rather than simply correlations, are difficult to establish in real-world populations using epidemiological methods. But because white nose syndrome had spread to some counties while the bat populations in other counties were still healthy, it provided a unique situation for an experiment that otherwise would have been unethical to conduct. He was able to look at other factors that could have potentially accounted for the increased infant mortality rate, but found that the increase in pesticide application was the only factor that had changed. In addition to consuming insects, bats play an important role in ecosystems by pollinating plants and dispersing seeds. Bats are the primary pollinators of wild bananas, agaves used to make tequila and mezcal, and many important fruit-producing trees such as mangoes. Bat populations being decimated by white nose syndrome can also be seen as a sentinel event for human health. While a lot of funding was allocated in past decades for antibiotic research, antifungal research has largely been ignored. As human healthcare providers, we have very few tools in our toolbox when a patient has a fungal infection. If resistance develops to the few antifungal agents we have, we would not be prepared to respond to an antimicrobial resistant fungal epidemic. Currently, we don't have any licensed human vaccines to protect against fungal infections. As we've seen with bats, A fungal pathogen spreading through an immunologically naive population can be devastating. Here's discussion questions to wrap up this third and final One Health case study. How did humans facilitate the spread of white nose syndrome? What changes to human behaviors could mitigate the spread going forward, and what actions could be taken to protect bats from infectious diseases in the future? How might surviving bat populations be different after white nose syndrome spread? Predict what changes animal species in the same ecosystem likely experienced as a result of the mass mortality among bats. How might climate change influence the spread of white nose syndrome? What other environmental stressors could interact with the fungi to worsen its impact?